Before we dive into the mechanics of a bioreactor and its operation, we should first examine the fundamental process we want to support in it. As we've discussed, our clonal cell line has been engineered to express our biologic protein of interest. The basic role of the bioreactor is to provide the care and feeding of the cells so that they make as much high-quality product as possible. So let's break down the essential elements in this process. We start with cells, and we want to make protein as a product. Protein synthesis involves a lot of cellular machinery, as we know, and maintaining cell viability and integrity will help with the conversion of the raw materials into the protein product. The cells themselves are made up of proteins, DNA, RNA, lipids, and sugars. These molecules consist of the basic elements of life. We will need to supply the raw building blocks that the cells can convert into these cellular materials through their metabolic processes. As the cells metabolize nutrients, they can also undergo cell growth or division. Since cells have a finite lifetime, we will need to understand the overall balance of cell mass during cultivation. The process of metabolism will also generate byproducts, materials that the cells can no longer use efficiently to build molecules or to source energy. Examples include ammonium ions, lactate, and carbon dioxide. The accumulation of these byproducts to toxic levels can inhibit the metabolism of the cells and slow production. Another aspect of our system is heat. Some metabolic processes are endothermic, while others can be exothermic. Most cells have evolved such that their metabolic reactions and structural integrity are optimal between 25 and 40 degrees Celsius. The bioreactor will need to control and maintain the optimal temperature for a given cultivation. In essence, a bioreactor is designed to facilitate the conversion of raw nutrients and materials into our protein of interest through a living system. As a result, we should strive for a close synergy between the biological and physical systems to maintain control and quality of the product during the cultivation. Let's now take a look at how we start to construct both the design of a bioreactor and a plan for its operation. In the simplest cultivation, we could add cells like these yeast into a container with media. We can appreciate immediately that this is not a homogeneous mixture. The cells settle at the bottom of the container and are in a dense environment. The consumption of nutrients here is not even, and it leads to a diffusion-mediated gradient inside the container. Similarly, oxygen diffusing from the atmosphere or the headspace of the reactor will not be transferred efficiently through the system. As a result, the cells won't have access to the nutrients that they need to make our protein. So let's mix the system by stirring. This added protocol increases the homogeneity in the system, but it also introduces additional factors like shear forces on the cells. We'll look at the finer points of how stirring affects oxygen transfer and cell integrity later, but for now, this idea makes it possible to expose the cells to a uniform environment throughout the reactor. We can now let this batch culture of cells work for us. Over time, the cells will take up nutrients and metabolize them. During this process, they divide at some average specific growth rate called mu. At the beginning of a batch culture, when the supply of nutrients is not limiting and the environment is favorable, the cells could grow exponentially. Meanwhile, some of their biological activity translates into product, our protein. This whole process looks great, except at some point the amounts of key nutrients available and the accumulation of waste products begin to impact the metabolism of the cells. Their growth and productivity reaches a so-called stationary phase. Cell death balances out cell division. Eventually, the rate of cell death exceeds that of cell division under limited resources, and the cell number decreases. Cell death and changes in metabolism that promote recycling of proteins in the media by proteolytic degradation also leads to a decline in total product. We should harvest the batch to collect our product before or just as we reach this end stage. You're probably already thinking about other ways to improve the operation of our production run to increase the yield of our product. We could add more nutrients to the reactor volume periodically to feed the cells. This process is called fed batch and is a very common approach. We could continuously add media with nutrients and simultaneously harvest media from the reactor containing cells, our protein, as well as the metabolic waste products. Or we could retain the cells in the reactor by some filtering mechanism while we perfuse media containing the desired product and cellular byproducts through the system. As you can see, all of these modes of operation can impact our production, though each has advantages and disadvantages that we will explore further later. Our examples here have emphasized controlling how nutrients are provided to the cells, but you can imagine that there are many other parameters that we need to pay attention to as well. For example, we will need to monitor and adjust the pH and temperature of the culture, and the partial pressure of oxygen supplied to the reactor. In many cases, all of these parameters are, by themselves, important to the cells, but the interrelationships between them and their combined effects can be complex, making the job of designing a cultivation process just that much more challenging. In the next segment, we will review the metabolic pathways that allow cells to survive and reproduce while making our biologic medicine. We will then consider how the bioreactor can provide a physical system to facilitate these essential processes.